Taxonomy is the formal science of classifying living organisms into structured groups based on their shared characteristics. A major challenge in biology is the use of common names. These names vary significantly by region and language, leading to confusion. Taxonomy offers a standardized system. By using rigorous criteria, it creates a universal language that allows scientists worldwide to categorize organisms without ambiguity. In 1735, Carolus Linnaeus published Systema Naturae, which established the foundational framework for biological classification used for centuries. Linnaeus organized life into specific hierarchical levels, moving from the broadest category of kingdom down to the specific unit of species. In the vast field of taxonomy, the species remains the foundational unit of biological classification. It serves as the primary reference for identifying and distinguishing individual types of organisms. A species is characterized by organisms that share fundamental traits. While every individual has unique variation, they belong to the same species because they share a common genetic and structural identity. This shared identity allows scientists to reliably distinguish one species from another, creating the consistency needed for global biological research. Phylogenetic trees are powerful diagrams that illustrate the evolutionary history of living organisms, showing how different species branched out from common ancestors over millions of years. At the base of the tree is the common ancestor. As we move upward, the tree branches, signifying points where lineages diverged due to evolutionary pressures. Taxonomists now use advanced genomic data to refine these trees, replacing subjective physical observations with precise molecular evidence of shared heritage. In 1866, naturalist Ernst Haeckel revolutionized the tree of life by identifying a critical gap in the existing two-kingdom system. He proposed a new kingdom, Protista, to house unicellular organisms that defied simple categorization as either plant or animal. Haeckel later introduced the kingdom Monera, specifically for organisms like bacteria that lacked a distinct membrane-bound nucleus. This marked a deeper understanding of cellular complexity, shifting taxonomy from mere observation to the study of internal cellular structures. Whittaker drew a clear line between the simple, non-nucleated prokaryotes in the kingdom Monera and the complex eukaryotes. He then divided the eukaryotes into four distinct kingdoms: Protista, fungi, plantae, and animalia. The model is most famous for the formal elevation of fungi to kingdom status. By recognizing their unique absorptive nutrition rather than photosynthesis, he corrected a long-standing error in plant-based classification. Standard biological classification, represented by the tree of life, organizes cellular organisms into three distinct domains: bacteria, archaea, and eukarya. However, viruses do not fit this model. Because they lack cellular structures and independent metabolic processes, they are excluded from the traditional tree of life. While cellular life maintains complex internal machinery, viruses exist merely as genetic information encapsulated within a protein shell, separate from the continuity of cellular lineages. Modern classification has moved beyond simple physical observation. Historically, scientists relied on subjective morphology, looking at shapes through a microscope, which often led to errors. Today, we rely on molecular genetics. By analyzing nucleic acids and protein sequences, we gain objective, quantitative data about evolutionary relatedness. Instead of guessing based on appearance, we use quantitative metrics. We compare gene sequences to calculate precise percentages of similarity between organisms. This genetic approach allows us to construct phylogenetic trees that map evolutionary history with unprecedented accuracy, transforming how we define the web of life. In the 1970s, Carl Woese and George Fox revolutionized our understanding of life's ancestry. They moved beyond physical appearance to the molecular level, specifically analyzing ribosomal RNA. By comparing the sequence of small subunit ribosomal RNA, they found a way to measure the genetic distance between all living things. This acted like a molecular clock. Before this work, scientists mostly focused on the split between prokaryotes and eukaryotes. But Woese found that archaea were fundamentally different. This analysis revealed the three-domain tree: bacteria, archaea, and eukarya. This structure highlights that archaea represent an independent evolutionary lineage. Traditionally, we view evolutionary history as a branching tree, where species diverge from common ancestors. However, horizontal gene transfer allows genetic material to jump between unrelated species. Because of these connections, scientists now visualize evolution as a web rather than just a tree. In modern clinical microbiology, identifying a pathogen is not always straightforward. When standard bacterial or fungal cultures return negative, physicians face a critical diagnostic impasse. Take the case of the patient known as Cora. Presenting with neurological symptoms like severe stiffness and involuntary movements, her standard laboratory cultures returned empty. This indicates the culprit is likely an atypical organism, a virus, or a non-culturable pathogen. When cultures fail, doctors must pivot. Molecular diagnostics, such as polymerase chain reaction (PCR) and genomic sequencing, allow us to detect genetic traces of a virus or atypical pathogen that would never survive or replicate in a petri dish. By moving beyond traditional growth-dependent methods, we can identify these hidden pathogens, potentially saving lives where standard diagnosis fails. In microbiology, identifying the species is often just the beginning. A species describes organisms with a shared core genome, but within that species exist subtypes called strains. Why does this matter in medicine? Because strains can have vastly different traits. One might be harmless or even beneficial, while another might cause severe disease. These differences arise through genetic variation, mutation, or horizontal gene transfer. Even within the same species, a strain that acquires new virulence genes can transform from a harmless resident into a significant health threat. Escherichia coli serves as a perfect example of why the species level of classification is sometimes not enough. While we identify them as a single species, their genomic diversity creates distinct strains. Most strains are commensal, existing as essential residents of our digestive system. However, specific pathogenic strains like O157H7 carry genes that cause severe foodborne illness. Precise classification is not just academic. For a clinician, identifying the specific strain is critical for diagnosis and determining the necessary treatment protocols. First published in 1923, Bergey's manuals have become the definitive reference for prokaryotic identification. We distinguish between two primary versions, the determinative and the systematic. The determinative manual prioritizes phenotypic traits for clinical ID, while the systematic manual bridges clinical observation with modern genomic and evolutionary data. In clinical laboratories, this manual acts as the bridge. A sample is taken, cross-referenced with Bergey's criteria, and leads to an accurate identification of the prokaryote. Many bacteria look identical under a microscope. Visual observation alone is insufficient for precise identification. We utilize biochemical tests to detect unique chemical markers. These identify specific metabolic pathways like sugar fermentation, which vary between species. Serological testing provides another layer of precision. We use antibodies that bind to specific surface proteins, creating a definitive immunological signature. By shifting from visual inspection to biochemical and serological evidence, clinical microbiology achieves the diagnostic accuracy required for patient care. Common names can be highly ambiguous. They often describe superficial traits, but fail to provide a universal reference, leading to confusion across different languages and regions. Scientific nomenclature has evolved. While early systems were strictly Latin-based, modern conventions incorporate Greek roots for precision and may even honor the researcher who discovered the organism. By adopting a strict structure of genus plus a specific epithet, scientists ensure a single, universal identifier. This precision is the cornerstone of biological classification, allowing for global collaboration without linguistic barriers. Haloquadratum walsbyi serves as an excellent example of modern taxonomic logic in nomenclature. Its name breaks down into three distinct identifiers. Halo denotes its extreme salt habitat, quadratum highlights its unique square cell morphology, and walsbyi honors Anthony Edward Walsby, who discovered this organism. In scientific literature, precision and efficiency are paramount. We begin by stating the full binomial name, such as Escherichia coli, in its entirety to establish identity. However, repeating long names becomes redundant. Therefore, after the first mention, scientists follow a standard rule. Abbreviate the genus to its initial, followed by a period. This transforms Escherichia coli into the recognizable E. coli, a widely accepted shorthand that maintains scientific accuracy while streamlining communication in charts, reports, and clinical notes. This practice prevents clutter in technical documents and helps focus on the specific strain or species characteristics being analyzed. Modern clinical diagnostics have undergone a revolution, shifting from slow, culture-based methods to rapid, high-precision genetic sequencing. Traditional culture-based methods require time for bacteria to grow. However, many pathogens remain unculturable, leading to diagnostic mysteries. In contrast, modern genetic sequencing identifies pathogens directly from patient samples, identifying hard-to-grow organisms in hours rather than days. By bypassing the need for growth, clinicians can identify viral or atypical pathogens that previously would have eluded standard detection, drastically improving patient outcomes. This technological shift turns diagnostic uncertainty into precision medicine. We have reached the end of our journey through microbial taxonomy. Our systematic approach has evolved dramatically over centuries. From the observational traits used by Linnaeus to the cellular complexity of the five-kingdom model, and finally to the molecular rRNA focus of the three-domain system, we have come far. Today, we no longer see evolution as a simple branching tree. Because of horizontal gene transfer, we now visualize life as a complex, interconnected web. Microbial taxonomy remains a fluid, living science, constantly refined by new genomic data. This systematic approach ensures we keep pace with the complex relationships within the microbial world,